Yeah. So we will start this. Good afternoon, Dhruv. Yeah, Kiran, only I had. Okay. So let us start the class, guys. In the last class, we have started about the microbes in human welfare, right? Today is the last part of this. I'm audible, right? Can you tell me whether I'm audible and screen is visible, right? PPT is visible and I'm audible, right? Okay. Thank you so much. So let us start. In the last class, what are the things we studied, guys, in this chapter mainly? So I have told you in this chapter mainly the products and the bacteria name, microbes name they will ask mainly. So here we have completed with microbes in the household product, microbes in the industrial product, and microbes in the sewage treatment. How in the sewage treatment the microbes are used, we discussed it in the last class, right? So in the last class, the primary treatment, primary and secondary treatment. Secondary is the biological we studied, right? In the secondary treatment, mainly we will use the microbes, right? Mainly in the secondary treatment, we will use the microbes, right? Okay. Then we studied about the biogas. Biogas mainly due to the production of the biogas mainly is due to the production, mainly due to the enzyme that is the methanogens bacteria, right? That is the archaebacteria we studied in the last class, that is present in the ruminants of the cow, right? Cattle. So then we studied about the microbes as a biocontrol agent. So here we studied that instead of using the chemical as a pesticide, we are using a natural predator for the controlling the pest, right? So that's why we called as a biocontrol agent, right? And in the today's class, we are going to study about the microbes as a biofertilizers. Okay, fine. So here you can see, yeah. So here is the first question, guys, for you. You can solve this if you studied whatever we did in the last class. Okay. So here they are given the microbes in the one column and in the list of that products in the another column. Okay. So you have to match both of the things. Streptococcus, Trichoderma polysporum, Monascus purpusis, Aspergillus niger. Which one is the correct answer? Can you go through this, match the following? I need everyone to answer to this question, all are in the class. Okay? I will launch the poll once again. Go through that before launching the poll. Streptococcus, Trichoderma, Monascus purpusis, Aspergillus niger. These are all the things we studied in the first class. Okay, I am launching the poll. You can answer me in the poll, guys. Very good, Saho. Correct. Very good, very good. Only 42% given answer. What about others? See the question on the screen and answer to that, guys. Answer me. Very good, Dhruv. Correct. Yes, what about others? Only three members have answered to this question. What about others? What about Kiran? What about Sanjana? Very good, Kiran. Yes, I'm ending the poll. Sharing the result, 75% given A and 25% given C. Yes, correct. So the correct answer is option A. Okay, so here we can match this guy. Streptococcus. Streptococcus will form the streptokinase, right? Streptokinase, right? Streptokinase, and the streptokinase, we all know that it is acting as a clot buster, right? Then coming to the trichoderma polysporum, what is the trichoderma polysporum is doing? Cyclosporine, right? It is a immunosuppressant, right? For the B, it is 3. And Monascus perfusus, it is produced, it is a yeast, right? It is a yeast. It will produce the statin. Statin lower the cholesterol. Yes or no? Then coming to the Aspergillus niger, it will produce the citric acid, right? So that's why the correct answer is option 1. And here I just want you to make a note of this, guys. Streptokinase, it is produced by the streptococcus. It is a clot buster. If any clot is there, it is going to be busted by this clot buster, that is a streptokinase. Okay. Then cyclosporin A, that is a Trichoderma polysporum, that is fungus. Kindly remember, it is fungus and it is a immunosuppressive. During the organ transplantation, we will use this cyclosporin. Then coming to the statin, it is obtained from the Monascus perfisus yeast. It is yeast. Don't confuse with the fungal. It is yeast. It is used for the cholesterol lowering in the heart patients. Okay. They may ask any type of the question because this is a very, very easy topic in your 12th syllabus. Okay. So in the last class, what are the things we studied, guys? Microbes as a biocontrol agent. So what we are doing here, instead of chemical, instead of chemical pesticides, chemical pesticides, we are using natural predators, right? This natural predator will feed on this prey, right? So that's why if controlling the aphids, we used the ladybird beetle, and for controlling the mosquito and mosquito larvae, we used the dragonfly, and to control the butterfly caterpillars, they will harm the cotton plant, right? For that, we used the Bacillus thuringiensis. What we did in the last class, we taken the spores, we taken the powder of Bacillus thuringiensis, we mixed it with the water and just poured on the plant, right? So thereby, whenever the butterfly and caterpillar feed on that plant, what will happen? So when that chemical went into when that Bacillus thuringiensis bacteria went into the gut of this butterfly, here what will happen? The butterfly feed on it will feed on BT. Just I am taking BT, okay, Bacillus thuringiensis. Now what will happen? This Bacillus thuringiensis will go to the gut, okay, mainly midgut, okay. When it go to the midgut, it will become activate. How it is going to become activate? Why it didn't activate in the stomach or in the saliva? Means it need, for the activation, it needs alkaline condition. Okay. So when it is activated in the gut, it started to make a pores. You all know that pores means holes. Holes in midgut. Holes in midgut. Okay. So when it is started to form the pores in the midgut, then so here what will happen? Osmolysis will happen. Osmolysis. Osmolysis will happen and it will burst. It will burst and it will die. Okay. So kindly remember this. Okay. If bursting means the organism that butterfly is going to be burst. No. So here the midgut become pores, right? So there is a water and salt imbalance will happen. Due to that, the butterfly will die. Okay. Do you understood this? How the Bacillus thuringiensis will work? For its activation require alkaline condition. Remember. Okay. Fine. And there is another thing we studied, that is a root pathogen. So here it is naturally present in the roots. Okay. It is naturally present in the roots of the higher plant. So what it is doing is, it will just check the pathogens. Okay. It will check the pathogens. It will cause the disease. No. If any pathogens it is causing the disease means, it will release some of the chemicals. Due to that, the organism will die. Pathogens will die. Do you understood this all the thing? We studied this in the last class. Can you give me thumbs up or any reactions, guys? Do you understood this? This is about the biocontrol agent, natural predators we studied in the last class. Some are natural predators, but some are not natural predators. Okay. Okay. So why we are actually using the natural predators, guys? In the last class, I told you. Why we are actually using the biocontrol agent, not why we are not using any chemicals here. Okay, 1 minute. One minute. Can anyone tell me why we are using the natural, why we are using the biocontrol agent rather than the chemicals? Can anyone tell me? So what I told you now, kindly see here. If you not understood Bacillus thuringiensis, how it is acting means, kindly see here what I told you. See here what is happening. So this plant, so the plant Bacillus thuringiensis powder will be there. Kindly see here. Powder will be there. That we will mix with the water. Okay. Then we will spray. We will spray that to the plant. Okay. When we will spray this to the plant, on the leaf and all the thing, there will be a Bacillus thuringiensis will be there. Right. On the leaves and everywhere, you can see that Bacillus thuringiensis will be there. So now what will happen? The butterfly come and it will feed on this. Feed on leaves. Okay. When the butterfly feed on the leaves, so now what will happen? The leaves is containing Bt, right? Bt, Bacillus thuringiensis bacteria. Right. So now what will happen? In the butterfly, midgut. Okay, the butterfly midgut was reached. Okay, the Bt is reached to the butterfly midgut. While eating our food, naturally will go to the midgut, right? Like that, when the butterfly eat that leaves, right? That leaves consisting of the Bt, right? Now that Bt will go to the gut from the mouth, then stomach, then it will go to the gut. Okay, in the midgut, what will happen? The midgut is having the alkaline condition, alkaline pH, right? So in the midgut, in the alkaline pH, the Bt will become active. Bt will become active. When it will become active, it is started to form the pores. It is started to form the pores on midgut. Midgut. Okay. So due to this pores in the midgut, it will make the osmolysis burst. Osmolytic burst. Okay. Due to this osmolytic burst, the butterfly will die. Butterfly die. Do you understood now? How many of you understood? Yes, yes, yes. Yes, Kiran. Kshetra, do you understood now? Yes. So why we are using the biocontrol agent instead of the chemicals means for the healthy crop. Okay. The crop should be healthy. For example, if you see, if we are using more chemicals means it will harm to the humans also. Right? Not only to the humans, all the other organism is going to be affected. But what we are thinking, we want to affect only the disease causing pathogen, right? Not to the others, right? So that's why for narrow sense, for affecting only, for targeting only for the disease causing agent, we are using the biocontrol agent. Okay. So it will not affect the other thing. It is working in a narrow spectrum. Narrow spectrum. Okay, fine. So this is what we studied in the last class. Then coming to the baculoviruses. I told you in the last class, we will continue this in the next class. The baculoviruses are the viruses. Okay, these are the viruses. These are belongs to the special family, that genus we called as nucleopolyhedrovirus. Kindly remember, Sanjushri, I will explain you once again in the last part, because I explained 2 times now, that's why. Okay, no problem. From now I am starting. Once again, I will explain you in the last, Sanjushri, okay? Just only some topics are there. I will explain you. Okay. So this is the first we are going to start today. The baculoviruses are the viruses actually, okay? So we are taking the viruses to control the insects and the arthropods, okay? So just imagine why we are not using the any chemicals for controlling the arthropods. Just imagine, so many insects are involved in the pollination, right? So if we just if we are targeting the only insects mean, it will kill all the insects, right? So we know that so many arthropods are involved in the pollination. So that's why we are only targeting the only insect and some arthropods. So that's why we are taking the baculoviruses. Okay, these baculoviruses belongs to the nucleopolyhedrovirus. Kindly remember NPD. NPD. Okay, nucleopolyhedroviruses genus. Here it will attack only the insects and other arthropods. Okay, so that's why we are using this baculovirus. And here you can see that why we are using this means it is a species specific and narrow spectrum. So that is what we actually required for the baculoviruses and the biocontrol agent, right? It should be narrow spectrum. If it is a broad spectrum, we will not use that, right? It is a narrow spectrum. It will target only the species specific and it will kill that. And it is insecticidal applications and desirable in the IPM. So what is actually IPM means? Integrated pest management. Integrated pest management means that is a program that is involved in conserving the beneficial insect. Can anyone tell me which are the beneficial insects, guys? No, any beneficial insects? Can anyone tell me?
ಅದನ್ನಂದ್ರೆ ಒಳಗ ಇದನ್ನಾದ್ರೂ ಅಂತ ನಾಳೆ
Can anyone tell me which are the beneficial insects? So insects which will do the pollination, very good. Bees, very good. Honey bees, right? The earthworm will not come. Earthworm will comes under the annelids. Yeah, only the arthropods. In the arthropoda, insects will come. No, that only we are taking. Okay, yes, honeybees and some of the insects involved in the pollination, right? Orchids and others, right? Butterflies. Yeah, some butterflies also. Yes. Okay. So we are not targeting those all the things. We are going to target only a particular insect which is causing the disease and which is getting harm for the which is harming the plants. So that's why we are going to conserve those beneficial insects in the program of integrated pest management. In this program, we are using the baculoviruses for the controlling the insects. Okay, kindly remember this baculoviruses that is belongs to the genus nucleopolyhedroviruses. It is works under the narrow spectrum. Okay, that's it, guys. So that is mainly we are using that viruses for controlling insects and arthropoda. That's it. Now we will talk about the biofertilizers. I think if any people are there who are farmers or having the well, farmers, children means you come to know that for the farming we require a fertilizers, right? So why we require the fertilizers, guys? Without fertilizers, can we able to grow the plant? We can able to grow the plant, but the growth of the plant will be very less, right? For that, we have to increase the fertility of the soil, right? So that's why when you go to the villages and all the things, they will do the crop rotation, right? What do you mean by crop rotation? If in this year they have grown some crops, means in the next year they will change that crop, right? That is called as crop rotation. So that will also increase the fertility of the soil. But here we are using a natural fertilizers, biofertilizers. Okay, so biofertilizers are natural. Okay, so these are the microbes are acting as a biofertilizer. It will increase it will increase fertility of soil. It will increase fertility of the soil and it will increase nitrogen content. It will increase nitrogen content. Nitrogen content. Kindly see here. First one is increasing the fertility of the soil by increasing the nitrogen content in the soil. Okay. Not actually for removing the weeds, increasing the content or organic content or the minerals in the soil. Increasing the mineral contents in the soil. Then only the soil will become fertile. not removing any pathogens or not removing any weeds and all the things. Okay, just increasing the minerals in the soil. When the minerals in the soil is increased, right, that we are going to call as fertile soil. Okay, do you understood now? Okay, so here, so let us understand biofertilizers. Biofertilizers. Bio, as the name is telling, bio means living. Fertilizers means living fertilizers we are using are the organisms that enrich the nutrient quality of soil. They are enriching the nutrient quality of soil. Nutrients means you all know that, right? Nutrients, magnesium, calcium, iron, sulfur, right? Phosphorus. These all are the nutrients. We require these nutrients for the growth of the plant, right? So, these biofertilizers or organism, microbes, these will enrich the nutrient quality of soil. Okay. Examples we are taking as a biofertilizers are bacteria, fungi, cyanobacteria. Kindly remember here, not viruses. Okay. Bacteria, fungi, cyanobacteria we are using. Okay. So, here just I want to tell you, see here, kindly make a note of this slide as it is because it will save your time. Okay. Kindly make a note of this slide as it is. Okay. Fine. When we take a biofertilizers, there is a bacteria, fungi, and cyanobacteria. Okay. First, we will talk about the bacteria, guys. Okay. In the bacteria, there are some bacteria are there that will work in the association, means that will work in association of the roots. Okay. So that we are going to call as symbiotic. Can anyone tell me what do you mean by symbiotic? Very good, Kshetra. Lichen. So lichen is a symbiotic association between fungi and algae. Very good, Redmi. Yes. So here you can see, first we'll talk about the symbiotic association, where two living organism is with the association, mutualistic association. Okay. So both of them is going to get the beneficial here. So here we will take an example, rhizobium. Okay. So rhizobium is going to attach to the roots of leguminous plant. Leguminous. In the so many MCQs, they will ask the question that which is the bacteria is with the symbiotic association with the leguminous plant. The example is rhizobium. For example, this is the root. Okay, this is the root. This rhizobium will form the nodules. This will form the nodules, and in these nodules, this will fix the nitrogen. This will fix nitrogen and the plant will get nitrogen. Okay. So these are the root nodules. The root nodules will be formed by this rhizobium. By that it will fix the nitrogen. The plant will get the nitrogen. The leguminous plant. Okay. Whereas Frankia is the one of the micro bacteria that will attach to the roots of non-leguminous plant. Non-leguminous. Kindly remember, in so many competitive exams, they have asked the question that which is the bacteria that is associated with the non-leguminous plant for the nitrogen fixation. Okay, that is Frankia. Frankia. Okay, so this is about symbiotic association. Means this will attach to the root, by that this will fix the nitrogen. But some organisms, some bacteria are free living. They will not attach to the root. They will not attach to the root. Not attached to the root. They are free in soil. Okay, they are freely living in soil, but they will fix the nitrogen. They will fix the nitrogen. In that, we are having two types. Okay, the bacteria which are there in the soil freely, but they are not attached to the any root. In that also we are having two microbes. One is a aerobic bacteria and another one is anaerobic bacteria. For the aerobic bacteria, we all know that it require oxygen. It require oxygen. For the anaerobic, not require oxygen. Not require oxygen. Right? For what? For fixing nitrogen. For fixing nitrogen. Okay. So the example is azobacter. The azobacter don't require the oxygen. This is the aerobic bacteria, and azospirillum and the Bijirkium. These require the oxygen for the fixing the nitrogen. Azobacter, azospirium and Bijirkium. So these are aerobic, require oxygen for fixing nitrogen. Then coming to the anaerobic, they do not require the oxygen for fixing the nitrogen. That is Clostridium and Bacillus. Okay. So now we understood the bacteria which increase the fertility of the soil. So one is with the association with the root, that is Rhizobium and Franklia, whereas another freely living in the soil without attaching to the any root. In that we studied aerobic, that is Azobacter, Azospirium and Bijirkium, and anaerobic, Clostridium and Bacillus. How many of you understood this? Are you clear with this topic? Are you clear with this topic, bacteria? Yes. So now we will go to the fungi. Okay. So we'll talk about the fungi. We all know that the fungi associate with roots of the higher plants, right? So here the mycorrhiza is association between root of the higher plant and the fungi. So here mainly two types is there, guys. One is a ecto. Ecto means what? So here is the root is there. Okay. So it is ecto. Okay. So just it will attach to the root. Just it will attach to the root. It will not go inside the root. It will not go inside the tissues. Okay. Ecto means attach. Attach to root only. Only root. Okay. For that example is the fungus name is Boletus and the plant name is Pinus. In the gymnosperm you studied, right? Pinus. For the Pinus root, the Boletus fungi is going to be attached. That mycorrhiza we are going to call as ectomycorrhiza. Okay. Then for some plant what will happen? This is the root. Okay. This is the root. So here it is attaching inside. Means it is attachment in the cortical cells. Attachment in the cortical cell into the tissue. Cortical cells in tissues. It will break the root and it will go inside the root where the cortical cells will be there, right? So there it is going to be survived. That fungus is the example is endomycorrhiza. The example for that is glomus. Glomus is the fungi, whereas the orchid is the plant. Do you understood fungi association, guys? In the fungi association, the association between fungi and the higher root, the example is ectomycorrhiza, Boletus with the pinus, and endomycorrhiza, glomus with the orchid plant. Do you understood this? Can you give me your reactions? Yes. So what about others? Lakshita, do you understood, pa? Sanjushri, do you understood, pa? Sanjushri, do you understood? So this is about the fungal association. Okay. So which one you didn't understood, pa? Sanju? Yeah, bacteria. Okay, fine. So the problem with you is, it is not actually, if no need to remember the bacteria what you studied in the cell chapter. Okay. So once again, I will explain you guys. See here. Okay, this is a revision type. Just I will explain you once again. So here what we are talking, the biofertilizers means what? It will increase fertility of soil. Fertility of soil, right? So mean it will increase the mineral content in the soil. Yes or no? Now we are taking the bacteria. In the bacteria, how it is going to increase the mineral content in the soil? In the bacteria, there are some different types of the bacteria are there. So how it is associated the plant is important. There are symbiotic bacteria. So these bacteria will attach to the root. Okay. These will be in association with the root. Okay. For that example is rhizobium. It will attach to the root. Thereby it increase the, directly it will give the minerals to the plant. Directly. Okay. Not in the soil. It will directly give the minerals to the plant. Okay. So that is symbiotic association. Directly. Direct. Okay. So here the rhizobium bacteria is there. So that will attach to the roots. Okay, that will attach to the roots. It will form the node-like structure. Okay, thereby it will give the minerals to the plant. Okay, so this rhizobium, it is going to attach only to the leguminous plant. Leguminous means, you know that groundnut, right? Groundnut is a leguminous plant. Groundnut. Okay. Then coming to the frankia is one of the bacteria that will attach to the root. So it will attach to the only non-leguminous plant. Non-leguminous. Non-leguminous plant. Okay. Leguminous plant means, I have told you example that is a groundnut, pea. These all will come under the leguminous. These are rich in protein. Okay. So some bacteria will not attach, but they are freely living in soil. They are free in soil. Okay. 1 minute. Yeah. So they are freely living in soil. Okay. So here in the soil, freely living bacteria, there are two types of the bacteria, guys. Okay. So one bacteria is like that. For the living, it requires oxygen. If oxygen is not there, means it will not live. It will die. Okay. So do you understood now? Aerobic bacteria means what? Aerobic bacteria means that will live only in presence of oxygen. Will you live without oxygen, guys? Can you tell me, will you live without oxygen? Tell me. No, right? How can we live like that? These aerobic bacteria, that is Azobacter, Azosporium and Bijerkium, require oxygen. In presence of only oxygen, it will increase the, in presence of only oxygen, it will increase the fertility of soil. Fertility of soil. Okay. Means it will increase minerals. Increase minerals like nitrogen. Okay. These all are actually nitrogen fixing bacteria, guys. In the environment, nitrogen is there, right? That is in the N2 state. Okay. So what we are taking the nitrogen is in the ammonia state. Right. So these bacteria will convert N2 to the ammonia, N2 to the NH3. So this is the main function of this bacteria. In the environment, in the air, nitrogen is there, right? We can't take that nitrogen. So for that, what they will do, they will fix that nitrogen in the soil. So N2 to NH3 they will convert. Later that, this NH3 will taken by the plant. When the NH3 is taken by the plant, obviously we will eat the plant, yes or no? We'll take the plant, yes or no? Or non-vegetarian only if you think. If they are feeding on the any animals means, obviously that animals also feed on that plant only, yes or no? By that, nitrogen is coming into our food web. Nitrogen we are taking by these bacteria only. Now you understood? So these aerobic bacteria require oxygen. Without that, it will not fix nitrogen. Whereas these anaerobic bacteria, these not require oxygen. Okay? These not require any oxygen. The example is Clostridium and Bacillus. Not require oxygen. Not require oxygen. Okay. So fungi, I already told you. The some fungi is nothing but the association of fungi plus plant root. Plant root. So if it is attached on the root, it is attached on the root outer side. We called as ecto. The example is Buletus, which is the pinus. Buletus is the fungi name. Pinus is the plant. Whereas the endoparasite, these are present in the tissues of the root. Tissues of root means cortical cells. Cortical cells. The example is glomus in the orchid plant root. Okay. So till now, do you understood? Sanjushri, now you understood, pa? Sanjushri, you are asking why these bacteria don't require the oxygen, right? Anaerobic bacteria, you are asking about why they don't require the oxygen, right? Means in the evolution, in the evolution, they grown without oxygen. In presence of the oxygen, it will become like the suffocating for them and it will die. Okay, they don't require oxygen, but they require some other gases. Okay, fine. Then coming to the cyanobacteria. Cyanobacteria we also call as blue-green algae. Blue-green algae, right? So here, this cyanobacteria, there are so many cyanobacteria are there, guys. Nostoc, and Anabenna. Do you know any cyanobacteria, guys? Can you name any cyanobacteria? Anabenna, Nostoc. So here, mainly Anabenna. Anabenna we are using, the powder of Anabenna, we are using for the paddy fields. In the paddy field, you know Godi, right? Godi in Kannada. Godi. Godi ಬೆಳೆಯೋದಕ್ಕೋಸ್ಕರ what we will do as a fertilizer for paddy fields, we will use this Anabenna fertilizer. Anabenna as a biofertilizer. Do you understood this? Very good, Kiran. Yes. Oscillatoria. Very good. Oscillatoria is also the, yes, Oscillatoria is also the cyanobacteria. Oscillatoria. Okay, any other? Nostoc. Very good, Sanjana. Yes, Nostoc, Anabenna, Oscillatoria we studied, right? Okay, so this is about biofertilizers. And one more thing I have to tell here about the mycorrhiza. Yeah, I told about mycorrhiza, right? It is a symbiotic association of fungus, right? Glomus we studied. Glomus in the orchid plant we studied. That is the endo mycorrhiza, right? So actually, what is the fungus is giving? Fungus, we know that fungus is getting the shelter and the food from the plant. Actually, what is this fungus is giving to the plant mainly? What is the fungus is giving to the plant means it will absorb the phosphorus. Kindly make a note of this. The fungus will absorb the phosphorus and it will give it to the soil and it will pass us to the plant. Okay. Then it will give the resistance to the root-borne pathogens. It will give resistance to the root-borne pathogens and also to the tolerance to the salinity and the drought. For example, the plant is growing, right? So there the salt concentration is increased suddenly. If it is not with the symbiotic association with the mycorrhiza means the plant will dry up. The plant will dry up and finally it will die. Right? But the fungal association along with that mycorrhiza means what it will do with the fungal association, it will tolerate that salt concentration, salinity, and also to the drought. Okay, waterless condition. Okay, and it will give overall increase in the plant growth and the development. The first one is phosphorus and second one is resistance to the diseases and resistance to the tolerance to the salinity and the drought, and the fourth one was the increase the plant growth and overall development of the plant. So mainly remember absorption of the phosphorus. So here you can see. So this is how the roots of this the mycorrhiza will look like. Okay. It is not actually very specific. Okay. So then we studied cyanobacteria. Cyanobacteria are autotrophic. Like how the plants are there, right? How the plants are autotrophic, right? So they will use the sunlight and they will prepare the food, right? They will prepare their own food. Prepare own food by using sunlight, right? So like that, the cyanobacteria also. So this will fix the nitrogen. This will fix the atmospheric nitrogen. I have told you already. In the atmosphere, N2 will be there. It will fix that into the NH3. Okay. So this is done by the anabenna and the nostoc and the oscillatoria. So here you can see, guys, this is the nostoc. Okay, this is the nostoc here. So this here we can call it as heterocyst. Okay, see here you can see that. Here, this labeling part, we were called as heterocyst. What is this heterocyst means? It is the place, okay, it is the place where nitrogen fixation will happen. Nitrogen fixation. Kindly remember, in the nostoc, heterocyst will be there, heterocyst. This is the nothing but heterocyst, okay. There's a place, heterocyst. In this region, mainly, the nitrogen is going to be fixed. N2 is going to be fixed. Okay, fine. And I already told you in the paddy field, cyanobacteria serves as a biofertilizer. Okay, and it also adds the organic matter to the soil and increase its fertility. Not only the inorganic matter like the phosphorus, sulfur, nitrogen, it will also add the organic matter to the soil to increase the fertility. Okay. So this is about the biofertilizers, guys. This much about this chapter. We have completed this chapter. Okay. So do you have any question regarding this chapter? Do you have any questions regarding this chapter? Okay. So if you don't have a question, means I have a question for you. The question is, identify a microorganism which is responsible for production of a immunosuppressive molecule, cyclosporin A. This all question is asked in the 2024. Tell me. Very good, very good, very good. Yes, yes, Kshetra, correct. Yes, the correct answer is. Can you tell me in the chat box, guys, the correct answer? Very good, Kshetra. Very good, Sanjushri. Very good, Rakshita. Yeah, very simple, very easy, right? The correct answer is option A, Trichoderma polysporum, right? So, from which organism that the Trichoderma polysporum that is acting as an immunosuppressive drug, that is a cyclosporin A. The organism name is Trichoderma polysporum. It is a fungus. Remember. Okay, next question. Which of the following is put? Which of the following is put into the anaerobic sludge? Anaerobic sludge digester for the sewage treatment. So let us see how many of you will answer to this question. Very good, Kiran. Very good. What about others? Very good, Sanjana. Get. Very good. Very good. So other than anaerobic sludge, where we using? Very good, Sahu. Other than anaerobic sludge, where we are using this Anna again? No, no, no. Think once again. Effluent from the primary treatment, we will send to the aeration tank. Yes or no? No, 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 guys. No, no, no. The correct answer for this question is option D. So kindly see here. So the sewage water. all are giving the wrong answer. Kindly see here once again, guys. I just want to complete this topic. Sewage water. For the sewage water, for the treatment, we have primary and secondary, right, guys? For the primary treatment, what we will do? First, we will remove floating debris, right, by the filtration, right? Then we will send this water. Then we will remove pebbles, right? So this is called as a sedimentation, right? So now we will get the water. After the sedimentation, right? So this is called as a primary sludge. Primary sludge. Yes or no? Primary sludge. Whereas the water above this, this is a sedimentation tank. Sedimentation tank. Right? So water from this, we will send to the large aeration tank. This is called as aeration tank. Yes or no? So, in the aeration tank, we are going to add a flocks. Yes or no? Flocks. These flocks consisting of bacteria plus fungi. Right? So, what is this flocks is doing? What is this flocks are doing? It will decrease the BOD, biological oxygen demand. Yes or no? So, after that, we will send this water to the settling tank. Settling tank. In the settling tank, what will happen? All the flocks is going to be settled down. These are the flocks. These flocks settle down. These flocks we are going to call as activated sludge. Activated sludge. And the above water is there. So these we will send to this is the effluent. This water we will send to the, yes, where? For the river, for the water bodies. Right. And this is activated sludge. We will use it as a inoculum, and also we will send to the anaerobic sludge. Do you understood now? So for the anaerobic sludge, what will happen in the anaerobic sludge? So these all are utilized and it will produce biogas. In the anaerobic sludge, biogas is going to be formed. Yes or no, guys? We studied in the last class. So now you understood? Remember, sewage water. Okay. So that is activated sludge. The effluent from the primary treatment, we will send to the aeration tank. Okay. Fine. So now let us go to the what triggers the activation of prototoxin to the Bt toxin of Bacillus thuringiensis in the bolo worm. Tell me, guys. So what triggers the activation of prototoxin of Bt toxin? I told you. So that Bt toxin is going to be activate in the midgut only, in the gut only, not in the stomach. Right? That is going to be activate. Why? What triggers? Very good, Dhruv. Very good, Kiran. Yes. Very good, Kshetra. Yes. What about Sahu? Correct, Sahu. Correct. Yes, JNV Baitang. Correct. C. Alkaline pH. Alkaline pH is going to be activate the prototoxin. I will tell you once again, guys. Prototoxin means inactivated. Inactivated toxin. Okay. Prototoxin means inactivated toxin. Sanjushri, I am explaining you this topic, but kindly see once again. You have asked me to tell me once again. I am telling you. So here what we are doing, BT powder we are taking. We are going to add that with the water. Then we are spraying. That. Do you understood, Sanju? We are spraying this powder with water. We are just mixing that water with the powder. Okay, BT powder means that is bacteria powder. Bacillus thuringiensis powder will be there. Okay, so that we will mix with the water and just we will spray that to the plant. Now what will happen? Now the plant is covered by this Bt toxin, Bacillus thuringiensis. Okay. So now bolo worm will come, or the butterfly will come, bolo worm. Okay. So now bolo worm will come and it will feed on the leaf. It will feed on the leaf. Okay. So now what will happen? That leaf particle they are eating, right? That will go to the stomach, then it will go to the gut. Okay, the gut. So till going from the stomach to the gut, it is inactive. It is inactive. That is in the prototoxin form. Okay. So, but when it is reached to the gut, it is having the alkaline condition, alkaline pH, basic pH, basic pH. So here in the gut, it will become activate. Okay. When it become activated here, activated, so just it will form. It is just started borrowing it will form the pores. Okay. It will form the pores in the midgut. Pores in the midgut. Okay. So it will lead to the osmolytic, osmotic lysis. So thereby it will die. So thereby the bolo worm will die. Sorry. It will die. Do you understood, Sanjushri? Sanju, do you understood, Pa? Okay. Fine. So once again, I want to tell you guys one more thing. Today is the last class, okay? So we are suspending the class from tomorrow. And once again, we'll start the class in the June. Okay? So we'll tell you in the groups to you when we are going to start the classes again. Okay? Today is the last class. I've launched the feedback poll to you. Kindly give me your valuable feedback. Summer holidays, Kshetra. You need holidays, right? Again you are going to study, again you are coming to school, again you have to study, right? Just you want refreshment, right? Yes or no? Thank you, Redmi. Okay, thank you so much, guys. You can give me your feedback. We'll meet after again in the June. Yes, Kshetra, but till now again you want some time for spending with your family, right? So many people are very happy. Don't know, Sanjana. For me also it is fine. If I continue the classes also, I'm very happy. I like to teach. I don't need any holidays. Thank you so much, guys. Miss you all. We'll meet in the June. And read once again, guys. Okay, whichever the. Thank you, Kshetra. I'm also going to miss you all. And once again, guys, when you are having the time, right, kindly read the chapter that is the inheritance biology and the molecular basis of inheritance. Okay. Thank you, Rakshita. So these chapters are very high weightage chapters. Okay, need more time to read also. Kindly go through that chapters and read well. Okay. Thank you so much, guys. Thank you, Kiran. Bye, Kiran. Have a nice holidays, guys. Yes, now then, from tomorrow, no classes will be there, Pa. The classes are suspended. And enjoy your time, guys. Yeah, bye bye.